Today, in this video, we are going to talk about molecular tools and gene analysis. The topics are separated into three segments. The first one is polymerase chain reaction, PCR. The second one is reverse transcriptase polymerase chain reaction, RT-PCR. And the third one is DNA sequencing. First topic we are going to talk today is about the polymerase chain reaction, the PCR. The PCR reaction is a method for making many copies of a specific segment of DNA starting with a very small amount. This technique can be used to identify specific microorganism from a small amount of DNA and to identify a person involved in the crimes from DNA on cigarette or in a single hair follicle. These are the gene of interest, and this one is this double-stranded DNA. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal. These are the gene of interest, and this one is this double-stranded DNA. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and the DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of the DNA using the polymer as the starting point. The temperature is raised to separate the DNA strands and then lowered sufficiently to allow the polymers attach. First topic we are going to talk today is about the reverse transcriptase polymerase chain reaction, RT-PCR. PCR can be modified into many modifications, including one called real-time PCR. Real-time PCR relies on the cell, the component of the PCR reaction. The target cell is the first one. The second one is the primer. The third one is the nucleoside triphosphate. And the fourth one is the Taq polymerase. This technique employs fluorescent oligonucleotide probe, which allows researchers to monitor the progress of reaction occur in the real-time PCR. The probe contains two fluorescent dye. One is the reporter dye, which is in the 5th prime, and the second one is the quencher dye, which is in the 3rd prime. When the reporter dye is excited by the light, it transfers its energy to the nearby quencher dye. This energy transfer is called fluorescent energy transfer. And last topic that we are going to talk about today is the DNA sequencing. DNA sequencing is the process of working out the order of the building blocks or bases in a strand of DNA. Before we can sequence the DNA, it has to be cut up into smaller pieces that are inserted into a plasmid DNA and then put into bacterial cells. This makes it possible to produce lots and lots of copies of it as the bacterial cells multiples. This also includes free DNA bases like A, C, G, and T. Modified DNA bases labeled with colored fluorescent tags are also added. These are called terminator bases, DNA primer, terminator bases, and DNA polymerase. It continues to add DNA bases until a terminator base is added.